which will be about food effects studies. We will have 4 lectures or 4 presentations. The first one will be about the bioequivalence of oral immediate release products under fasting and fit conditions. By Dr. Nilüfer Tanrı. Dr. Nilüfer works in the division director US FDA. So please start. Please, Dr. Nilüfer.
Okay, good afternoon everyone. We'll get started. My name is Nilüfer Tanrı, for those of you all who didn't catch my name earlier, and I'm with the U.S. Food and Drug Administration, where I serve in the role of the Division Director in the Office of Bioequivalence, which is in the Office of Generic Drugs. Now, before I proceed, I have a disclaimer here. Oops, sorry. I have a disclaimer here to read. It basically says that the views expressed here are my own views and should not be construed to represent the FDA's views and policies. So, having got that out of the way, here is what I'm going to be presenting. First, I'll start with an overview of the current recommendations for bioequivalence testing for generic and new drugs. And then I'll proceed to describe the progressive changes to the recommendations over time. And for that, I'll touch on the history and background so that you can see how our thinking evolved as we gain knowledge and experience in this area. And then I'm going to use some case studies from regulatory submissions to support our recommendations for asking for BE studies under fed conditions. And then finally, I'll wrap up the presentation with a summary. So let's take a look at the recommendations for BE testing for new drugs and generic drugs. I think first we need to stop and understand what is the goal of BE test assessment? Why do we do this testing? What's the objective over here? So BE studies are done primarily for formulation comparisons. So the objective here is to measure and compare the formulation performance of 2 or more pharmaceutically equivalent compounds. For example, a generic drug and a reference product. And I'd like you to keep this in mind. So one more time, BE testing, we are not demonstrating safety, we are not demonstrating efficacy. What we are looking for here is the formulation performance in terms of similar rate and extent of absorption of the drug. So as I take you through the slides, we need to keep this basic principle in mind. Okay, so I'll move to the new drugs. For the BE studies that are conducted for new drugs, they are really comparative bioavailability studies and not BE studies in the true sense. And that's because they are not required to strictly meet the 80 to 125% BE limits. And for oral immediate release new drugs, at pre-approval, just a fasting BE study is considered sufficient because we have of the totality of the information that is submitted and reviewed for these drug applications, because these are new drugs. And so these kind of studies, they are generally to bridge the formulations. For example, an early and a late clinical trial formulation, or sometimes the to-be marketed formulation can be a little different from the clinical trial, what was used in the clinical trials. And here we have some other examples. So those are the ones where we do the bridging. At times, we may need, the nature of the formulation change may be such that we may also ask for additional food effect study at pre-approval. Coming to post-approval, if the extent of the change is significant enough that we may need testing in vivo, in that case, generally just a fasting BE study would be sufficient. However, for generic drugs, at pre-approval, we ask for fasting as well as PET-BE studies. There will be some exceptions that apply, and I'll cover them in my subsequent slides. At post-approval, again, similar to the new drugs, we only would ask for fasting BE study. So I wanted to first talk about what's the difference between a food effect and a PET study. It sounds similar. So food effect are the food-drug interaction studies, and they are conducted to show the effect of food on the rate and extent of absorption of the drug when it is given shortly after a meal compared to a fasting state. And these studies, they have two purposes. First, the data from these studies are used to determine whether or not to conduct pivotal studies with regards to food. And then the second one, it supports labeling instructions for dose administration, again in relation to food. On the other hand, PET studies typically will be conducted performed for the generic drugs, and the comparator here is a designated reference standard or the reference listed drug. And the purpose here is to ensure bioequivalence under similar conditions, meaning the generic and the reference are both tested under PET conditions, and we want to show bioequivalence under those conditions. And the reason we are asking for that is that then we can be assured about the substitutability with the generic drug. Now we also ask that the test conditions for the fasting and PET-BE studies be standardized. So with regards to the meal, we ask for high-calorie, high-fat breakfast. And that's because, of course, we know that the fat content, viscosity, volume, etc., that affects the drug absorption, and in turn that can affect the safety and the efficacy of the drug. And a high-fat meal represents the most stressed meal condition. So we know that high-fat, high-calorie will have the greatest impact on the GI physiology, and which in turn will affect the absorption. So when tested under this condition, if the formulation differences do exist, then this test under these conditions is more likely for us to detect those differences. And that's why we ask for this high-fat, high-calorie meal. The other thing is we test between two extremes. One is no food, which is the fasting study, and then a highly stressed meal condition, which is this PET study. And when we do that testing, then we are assured that any combination, across any combination of meal conditions that in real life people may be using, this product will still perform and be bioequivalent. In terms of the timing relative to the dosing, for the fasting, we ask that the dose be given at least 10 hours after the last meal. For the PET study, it's within 30 minutes from the start of the meal, which is preceded by fasting for at least 10 hours. The strength is typically the highest strength which we ask for testing. We also allow for meal substitution. For example, we get many studies which are conducted in India, and many of them would be using vegetarian meal. That may be acceptable as long as that meal has similar calories from fat, protein, and carbohydrate as the FDA standard breakfast, and also the meal volume and the viscosity should be comparable. Now, in some cases where the studies are conducted in patients, in those cases the patient's treatment condition and the disease condition, and the treatment need and the disease conditions, we take those into consideration, and in those cases we may allow for a non-high fat meal provided there is a justification. For example, if the health condition of the patient raises safety concerns, or the patient cannot tolerate a high-fat diet. So like cancer patients, we generally would hear they cannot really tolerate a full FDA high-fat, high-calorie diet. And in those cases, we would allow. With regards to the new drugs, where we are talking about the bioavailability, US FDA rarely asks for effect of different types of meals on the bioavailability of oral drug products, oral IR drug products. So next I'll switch gears and I'll talk about how the recommendations progressively changed for the FED study, and we'll give you some history and background over there. So here, as I said, we generally ask for immediate release products. We still ask for fasting and fed for oral IR products, although some few exceptions apply. Now, prior to 2002, we would ask for data for the point estimates and the 90% confidence interval for the fasting study, but for the FED study, demonstrating just a point estimate to be within 80 to 125 was considered sufficient. However, in 2002, end of 2002, we posted our PK endpoint guidance, and in that we recommended additional statistical analysis for the 90% confidence interval as a condition of approval for the FED study. So just the point estimate alone was no longer sufficient from 2003 onwards. In addition to that, in 2011, we made further changes to our recommendations for the criteria or the categories which fall under the exceptions to conducting the FED BE study. Okay, so here's a snapshot of the exceptions to the FED BE study, and also you can see the changes from 2002 to 2010, and then we move later. So a FED study would not be required or would not be recommended, I should be using the word recommended, would not be recommended if the labeling states that the drug should be taken on an empty stomach, or the labeling makes no mention about the effect of food on the absorption or the administration. Just to clarify, labeling, we call it labeling in MR, you call it SMPC, so that's the thing what I'm referring to. So also, if the drug product, both the test and the reference are rapidly dissolving, they have similar dissolution profiles, and they contain a BCS class 1 drug substance. And the other category was if the study is done in patients at steady state, and they cannot, I just mentioned where they cannot tolerate the high fat for these patients, we don't upset their regular regimens. So if they are not taking their drug with the food, they would continue not to do that. So that's where when we do it in steady state, we don't specify whether it should be a FED study or a fasting study, unless there are certain safety issues. So in 2011, as I said, we revised the recommendations, and the main change was that irrespective of whether the labeling mentions the food effect or not, we are going to ask for FED studies on all immediate release products, unless there is a safety issue, that means there is some anticipated serious adverse events. And the other thing that was updated is that in addition to BCS class 1, we included the BCS class 3 drugs. So, sorry. All right. So what's the rationale for asking for the FED study? We know that the food effect results from a combination of several complex factors, and it's influenced by the quantity and the composition of the food. It's also influenced by the formulation and especially the excipients that can affect the bioavailability. And so, in our opinion, the magnitude and the direction of the impact of the food on bioequivalence, they are difficult, if not impossible, to predict without a FED-BE study. And so that was our reason for changing it. And also we find, and I will give you examples, or I will, in the case study, I'll show the data from the fasting study and dissolution testing is not always enough to ensure us bioequivalence under the FED conditions. And then the question always comes, but the labeling doesn't say that, so but why do you, FDA, why do you need it? And also I would say here, because we're talking about harmonization, EMA doesn't ask for FED studies under all conditions. There are very specific situations, at least in the guidelines, where they ask for, where they say they will ask for a FED study. So FDA is a little different in that respect. So why do we still ask for it? Because we find that relying on the labeling instructions to guide the recommendation is not always adequate. As I said earlier, how do these statements get into the labeling? The statements about the clinical relevance of dosing with food, they are influenced by how the drug was dosed with regard to the meal in the pivotal clinical trials. So those are the phase 3, phase 2, and phase 3 studies. Also, the food effect is not described for all drugs in the labeling. And so for many drugs, the information about the effect of food on the bioavailability is it's either not available, which means it wasn't tested at some point, or it's not included in the labeling, or it was tested, but at that time, the review division might have felt it's not clinically relevant, we don't need to put it in the labeling. So there has been inconsistencies, like how the information has gone into the labeling. And also the labeling language, we can see it's not always explicit, and it's not consistent, as I just mentioned, and it's also subject to interpretation. So for example, if you see in our labeling, an empty stomach would be described in multiple ways. So take it on an empty stomach, take it 2 hours before and 1 hour after, and some labelings were talking about take 1 hour before and 2 hours, it's the other way around. So it's sort of like, it's not very explicit. The other example that I can think about is if somebody says, if the labeling says, take under fasting condition. Alright, so somebody takes under the fasting condition, but in 10-15 minutes, okay, now I've taken my drug, now I can eat whatever I want. So in real life, there are different situations that can happen. And also, truly during the day, we cannot say that the stomach is under a fasting condition. Food is there in the stomach. And also, even though we say something in the labeling, it does not preclude somebody not following the labeling. And so we want to assure that the bioequivalence of the drug under all different conditions. And that's why we asked for this FED study. So giving you this background, I'm going to move to case studies. And here you will see you will see the value of asking for FED studies, although it's an immediate release product. So the first case, the background, so we look at the left side here, the background. It's just a conventional formulation of an immediate release oral product containing commonly used excipients. Now, I'm not able to reveal the drug name because of the confidentiality, but you still have all the information. So this is immediate release. It contains the usual, normal, commonly used immediate release excipients. In the labeling, there is no mention of the food effect. And when the bioequivalence testing was done, the test product met the BE limits for the fasting study. Now the issue here is that the food affected the Cmax to varying degrees depending upon the formulation composition. What I mean by that is, in the labeling of this drug, the components are mentioned. So we have multiple generic forms we submitted for this drug product. So since the components are mentioned, all the different generic applicants use the same components. The difference was that they used the amount of those components was different. Okay? And so here, the formulation had similar components, but they differed in the composition, specifically in the amount of MCC and starch. So here I've noted down 5 generic applications. And here is the reference product. This is the table showing just the MCC and the starch. And here you see, I just put in here the mid strength and the high strength. And we see those strengths are proportional, so the amounts are similar. And we see this starch about 60%, 27%. And look at the generics, the MCC 83, 81, 84. And then the last two, the MCC is a little lower. And this is what, this first end-up. The FED study did not meet the BE limit. So the red ones are the ones which did not meet the BE under the FED conditions. And these two, they were fine with the BE, FED conditions. So here is the statistical summary of the FED statistics. And you will see the first ANDA, the point estimate is 71, and this is so much on the lower side. This is a true, it looks like there's no other way but to reformulate this. This is not going to, even if you repeat the study, you're not going to pass it, just from the data. Here, the next two, we see the point estimates, this is marginally passing, but again, the 90% confidence intervals were out. Whereas for these last two, where the starch was a little higher, and MCC was lower, it was a little lower compared to the reference, and here we see that the FED study was acceptable. So when we look at the data, this is the TR ratio, test to reference ratio for the FED study and for the fasting study. We see there is a delay in the absorption, like the Tmax is longer. It's about longer by about 65%. And the Cmax was reduced by about 25% compared to this one. So it's not at 100. There is an impact on the absorption. Where, yeah, the AUCs were not an issue. And so here, in this particular case, the conclusion was that the amount of some excipients were not appropriately selected. And for the ENDA number 1, which was totally out of the range, the applicant themselves, they speculated that the food effect was related to a high proportion of the MCC, the microcrystalline cellulose. And their speculation was that because MCC swells, in presence of food, the barrier becomes even more, and so that was the reason for impacting the gastric emptying or slower release and an impact on the Cmax for this particular situation. So that's the first example. And I'll show you another example here. Again, IR product. This was conducted, this was a bridging BE study referencing an approved immediate release product. So this was what we call as a 505(b)(2) application. this particular drug, it is used for the treatment of acute pain, administered 4 to 6 hours interval as needed. This is a BCS class 1 drug. I would want to mention even the first example that I showed you. It's a highly soluble, highly permeable drug. In the labeling, the bioavailability of this drug is greater than 90% and high soluble. So my point here is it's the API characteristics. We feel, oh, it's a BCS class 1, does it matter? And that's why this is the example that I picked. So this is a BCS class 1. And again, it's a conventional IR formulation, nothing very specific, nothing fancy about it, dry blend and a direct compression. The issue, again, similar issue. The FED study met the BE criteria for AUC, but for the Cmax, it was approximately 20% lower. While the fasting study, it met the BE for all the PK parameters. And delay in absorption, which means the longer Tmax, the concern here was that the delay in absorption can result in a decrease in the efficacy because this is a pain treatment medication and it has to be given every 4 to 6 hours. So, looking into this application, when taken with food, absorption was substantially delayed. The median Tmax was 4 hours for the test product, where it was 1.5 hours for the reference product. The exposure over 4 to 6 hours dosing interval was lower, and I'll show you the plots for that. And it took longer to achieve comparable systemic exposure compared to the approved formulation. However, when it was taken without food, there was no delay in the absorption. The median Tmax was 1.03, and this one is 1 hour, very similar. The exposure at 4 to 6 hours was comparable, and I also put the resolution profiles here, and you can see the resolution profiles are also comparable. This first point is really at the 5 minute, it's a very early point to say, oh, they are not comparable. So that's with the fasting condition. And here, we have the TR ratios for the partial AUCs over the dosing interval. I just want to clarify here, we are not recommending partial AUCs for this drug. This is just a way of analyzing the data a little further when we see this kind of data. So the partial AUC against the time. And for the fasting study, we see that by 4 hours, like at least 90% is comparable to the reference. The test is comparable to the reference. But if you look at the FED study, even at 5 hours, it's just reaching 50% of the reference. And 6 hours, maybe 60 or whatever percent. So maybe about 60% of the reference. So, and this is a pain medication. This is something that we cannot allow. So this particular study was not accepted. The applicant was asked to reformulate this one. So the conclusions for this case study is that the excipients in the formulation likely affected the drug absorption in presence of the food. And the formulation, again, the change in the formulation was that they added additional gelling agents to the test formulation. So something similar to the first one where if it forms a gel and then under the FED conditions, the absorption gets affected. Excipient food effect interactions resulting in delayed onset of action can be clinically significant for this particular drug. And recommending drug administration and the fasting every 4 to 6 hours is not practical for patients. So the argument from the sponsor was, but in the labeling, we can just not have, the original labeling says without consideration for food, it can be taken. But when they reformulated, they were proposing to change the label, but that's not practical for a patient to be under fasting conditions every 4 to 6 hours. So that was not accepted, and we kind of rejected that study. So, yeah. So in summary, fasting BE studies are generally sufficient for new drugs, whereas for generic drugs, we ask for fasting as well as FET studies, unless there are some exceptions, especially if there are safety issues. And I showed you examples where interaction between food and drug formulations, they are complicated, it's affected by the component and the composition of the food, and the quantity and the component and composition of the formulation, and the quantity and the composition of the food. Excipients in conventional dosage form have the potential to impact BE under the FET conditions, and the impact on the gut physiology can influence the absorption of the drugs, including drugs which are BCS class 1 drugs. So with that, I'd like to acknowledge Dr. Connor and Dr. Landberger for giving me critical comments on my slides, and Dr. Mehta and Dr. Penny, they are on the new drug side, so I have to talk to them about what the expectations for the new drugs are. And thank you very much.
Thank you, Dr. Nilüfer, for the nice presentation about FET and fast conditions, the changes in the recommendation in US FDA, and the rationale behind that. I think we have time for one question only. Dr.
I wanted to ask, like the case you presented is, it is a case study, and it's such an extreme that even if it is a class 1 and yet the effect of food was demonstrated in the food effect. But can't we say that 90% of the cases, or maybe more, I don't know, it's just a question, where the food effect is really something intrinsic to the API itself, to the nature of the molecule, rather than effect of the formulation. So I'm not saying it's 100% thing, but it is something worth mention that the food effect is not really across the board for all the formulations.
For bioequivalence testing, we are testing generic drug and the reference drug. The API is the same formula. So that the food that you are getting may not be applicable for bioequivalence. It's mainly the recipients that will have to.
This is what I'm trying to say, is that the case we demonstrated was class 1, and yet the effect of excipients was such an extreme case. If we let this alone, okay, can't we say that the food effect is really something to do with the nature of the molecule, rather than, I mean, in the majority of cases? Why would I put a regulation to govern all the molecules, although it is more or less the intrinsic behavior of the drug moiety itself, rather than the formulation?
So I would say for APIs, yes, the poor solubility drug products may I don't know if I can say that. We have examples of this kind of so we do not test using why should we test, but just looking at the formulation, how are you going to determine that there will not be So we from the regulatory, I do not disagree that maybe this is something which could be revisited and looked at it in a little different way. But as a regulator, we cannot agree that if it's 5%, that's fine. So So if somebody has a pain and I'm going to disregard, knowing that this population is not going to perform for that pain, as a regulator can do that.
I think we have just one comment here, about the same subject.
Yes. Sattam Al-Dair, Spimar to Saudi Arabia. I just would like you to kindly elaborate more about the post-approval. You said if the product is already approved, we will ask during change only for fasting study. Yeah, can you please elaborate more about that and please confirm if this is mentioned clearly in the guidelines?
The last reference, the super IR guidance, it's mentioned in that one. You can see that. So it all depends upon the level of change. We have level 1, level 2, level 3. So if the change rises to the level of the level 3 change, only then we ask for an in vivo study. When we ask for an in vivo test capacity,
Yeah.
studies.
Please use the mic.
You guys want me to repeat? So right now, my topic is only on IR. No, so what about modifier? I don't know if you want to.
Maybe later on. One last question here.
I think we have, it's better to allow for the participant to have more frank discussion, but that to share the information between the presenter and the others. Otherwise, this is not the target for such conferences. I'm sorry to say that. It is a very important topic which we have really a lot of questions. Therefore, please allow for more time to
We
discussion.
will allow, I think after the end of the session, we will have more discussion because we will have also 3 other presentations.
I have two questions in your presentation. You mentioned that for the PS class 1 and class 3, this is not needed to do fit study. I don't find anything in the FDA, in the EMA, in any guidelines at all which state like that.
I would think
That's the first
I
question.
would think it's in our PK endpoint guidance, which was revised and the draft was posted in 2013. It should be there.
For IR products.
So we don't have a separate guidance for IR and MR for the PK endpoint. It's one guidance for generic drugs. And you will find it there.
No, I mean, I'm sorry, I'm confused. Is this to bioequivalence the bioequivalence study or to bioequivalence only the fit study?
Sorry, I didn't understand.
It means for all of us know that is for class 1 and class 3, you can bioequivalence the bioequivalence study. But this is what you mentioned, is to be bioequivalence the fit study only for the class 1 and class 3.
Can we?
Okay. Some other what you mentioned in your presentation, that is the fit study can be biowaived when the API is PCS class 1 or class 3. Okay? I means what is you can biowaive all the bioequivalence study when the board and the API is class 1 class 3, but not only the fit study. Why is the fit study?
Okay, so sorry. Are you saying, let me understand this. Are you saying because I had this up there for the PET study, that why are we asking for the testing study and that's not being made? Is that what you're asking?
Please ask yes.
Yes.
You want us to waive class 2 and class 4?
No, 1 and 3.
Okay, I'm sorry, can I just talk to you?
Later on,
Okay. The other point is that you mentioned that the partial area under the curve in one of the study is refused from the FDA. What about the coefficient variance? I mean, the FDA stated clear that is a CV in the distribution elimination phase must be not too high. Otherwise, you have not to use at all the partial the curve study.
Again, I did mention we are not recommending partial AUC for this drug product. This was just to slice and dice the data and look at it in all different ways. So this is just a way of presenting and looking at it at what point does it come equivalent, about 90%. Versus in the PET study, it goes up, it's not even up to 50% at that time. So we're not asking for partial AUCs. This is, I mentioned this very clearly at the beginning. We are just, this is just a way of analyzing the data from different angles.
Okay, thank you, Dr. Niloufer. Now coming to the second presentation, which will be by Dr. Louise Kikawi. She's managing partner at SCK Pharmaceutical Consulting and ex-US FDA assessor and ex-Saudi FDA consultant. She's going to talk about bioequivalence studies with immediate release products, administration conditions, and composition of these.
Can you hear me? I think so, because I can hear myself too. Thank you to the organizers of the conference for inviting me to present here today. It is truly an honor to be here for the third year. I think this is my third year here. And today I will be discussing the BE studies with immediate release products in terms of administration conditions and composition of meals. The good news is that this is the part of the program where we're going to make up some time, because a lot of what I'm going to discuss has already been discussed. The way my presentation has been structured is a little bit different. I'm going to do a little bit of a comparison between the US FDA and what EMA has been asking, and the reason why I did that is because a lot of the GCC and EMA region follow the EMA guidelines. So we're going to see the comparisons and what the differences or similarities are in terms of harmonization. Okay. Can you still hear me? So the outline of the presentation is as follows. I'm going to talk about BE studies for IR products as EMA and US FDA. Some discuss administration conditions, and then I'm going to talk about composition of meals, and I'll be giving some examples in this situation. So this is more what the guidelines say, and then maybe what you see in practice. I'm putting up my references now, so you know where I'm coming from and what references I've used. I have the EMA guidelines, I have the GCC guidelines for bioequivalence. I also have the US FDA guidelines for food effects, and also the bioequivalence study with pharmacokinetic endpoints. And this is, I think, the most recent guidance from the US FDA as it pertains to generic drugs. So in general, this is a depiction of what EMA and US FDA ask. We see that in EMA, it's typically one study, fasting study, 2A, single dose, whereas in the US FDA, there's for immediate release dosage form, you need 2 studies for fasting plus a fed state study, depending on certain specifications. So there's a lot of information on this slide from the US FDA guidelines, but I wanted to put it out there so that if you're working at your desk or you need to figure out fasting fed, you can just look at this presentation and be able to find all the information there. So for immediate release dosage form from the US guidelines for capsules and tablet, it recommends, as I said, a single dose fasting study and a single dose fed study, depending on certain situations. The exceptions are, as Nulifa already said, that the exception would be when the dosage and administration of the label states that the product should be taken on an empty stomach. So for example, if the labeling states that the product should be taken or should be administered 1 hour before or 2 hours after a meal. The other exception is for orally administered immediate release products. Products labeled to be taken only with food. Fasting and fed studies are recommended unless there's a serious adverse event with, which is anticipated under fasting condition. And Nulif already discussed this, especially with cancer patients who are not able to withstand the high-fat meal that is administered during fed studies. Some additional information from the US guidelines. In addition to a BE study and a fasting condition, it's kind of a repetition. The other one tells you the exception, and this tells you when you should do it. A BE study is recommended under fed condition for all IR products, with the same exception, the label. And here I should note that there's an update for BCS Class 1 and also BCS Class 3. So BCS Class 1 and BCS Class 3 drug products, you have a biowaiver option by relying on the BCS characteristics of the product. Now this means that you're waiving, which was one of the questions earlier, you're both waiving the fasting and the fed study, so you don't have to do any bio studies, bio, which means both, and you can rely on the BCS guidance and the specifications that are found in the guidance. So some clarification to some of the questions before. And if a product, take only on an empty stomach, then that's clear, very clear. So let's see what EMA says. In general, a bioequivalence study should be conducted under fasting condition, as this is considered, according to EMA, to be the most sensitive condition to detect potential differences between formulations. Now, as Nulifa had mentioned earlier, for this bioequivalence studies, we're doing a comparative study, and it's differences between the two formulations that are being detected. EU has decided that fasting is the most sensitive conditions. Earlier in the day, I think Anders was joking that we shouldn't ask why to the regulators, but I think Nulifa has provided many reasons why US FDA considered conducting the BE studies as a significant point. So according to EMA, food products where the SMPC, the label, recommends intake of the reference medicinal product, I think we should also harmonize how we refer to the products and how we refer to the labeling, and because it's very different. You can see the differences in the wording. Reference medicinal product on an empty stomach or irrespective of food, the bioequivalence study hence be conducted just under fasting conditions. For products where the SMPC recommends intake of the reference product only in the fed state, then the bioequivalence should generally be conducted under fed condition. So if it says under fed, you do the fed study and you don't do the fasting study. And that's the difference, the main difference between US guidance and the EMA guidance. And because we are in the NEMA region, GCC, this is kind of where we are in practice. Now, when we talk about harmonization, where do we want to be? Okay. Some other additional points if some formulations have specific characteristics such as microemulsion, bioequivalence studies performed under both fasted and fed conditions are required, unless the product must be taken only under fasting condition or only under fed condition. So here is a situation where EMA is requiring both the fasting and the fed study based on the formulation characteristic, or depending on the recommendation in the label or SPC, you can do fasting or fed. EMA also provides a recommendation on how the studies can be conducted. You can either conduct 2 separate two-way crossover studies, or you can do a 4-way crossover study. And I should ask the people who conduct the study if any of you have done a 4-way crossover study to show the fasting and the fed. I thought that was kind of interesting and different. So thus far I've given you the similarities and the differences in terms of fasting and fed studies and how they should be conducted for the 2 major regions. Actually, they're supposed to be timing myself, but hopefully I can make up some time. Now we're talking about administration condition, and Nulifar, again, has talked a little bit about this. So this is some repetition. So according to the US FDA, fasting BE study should be conducted after an overnight fast of at least 10 hours. Subjects should be administered the drug product with 240 mLs of water. No food should be allowed for at least 4 hours post-dose. Water can be allowed as desired except for 1 hour before and 1 hour after administration. So the administration conditions under fasting, this is how it should be. And then it says for the rest of the study, the meal should be standardized, and this is just to avoid introduction of variability. Now for the fed study, and this is where we are, following an overnight fast of 10 hours, which is similar to the fasting study, subjects should start the recommended meal, the high-fat meal, 30 minutes prior to administration of the drug product. The subject should eat this meal in 30 minutes or less. However, drug product should be administered 30 minutes after start of meal. So if you're a slow eater and you're participating in a BE study, at 30 minutes, you're going to try and, hurry up, you take the medication. In this slide, I also wanted to give an example of a study that I've encountered in the past where the study was conducted, it was a fed study, and the documentation from the fed study showed that some subjects ate the meal, did not complete it in 30 minutes, and so the subject had eaten, in the submission, it was like 40% of the meal or 80% of the meal, and they were very good in documenting how the subjects eat the meal, but if the subject doesn't eat the complete meal, how does this affect the results of the fed study? So then there was a decision, do we drop the subjects who did complete the meal? And if you include all the subjects, the study passed. But then when you started to drop the subjects who did not complete the meal, then the study really failed. It was pretty bad study, but this is a good case study where you have to standardize everything, and you have to keep within the times and the amount of food to be taken so that you can meet the caloric requirement that I'll talk about in the next couple of slides. Again, the drug product should be administered with 240 mLs of water, and no food should be allowed for at least 4 hours post-dose, and the same thing, water can be allowed, and standardized meal throughout the study. So what does EMA say with regards to the administration conditions in the fasting and fed studies? The subject should fast for at least 8 hours. In the US guidance, it says 10 hours. So here is a point for harmonization, okay? As fluid intake may influence gastric passage for orally administered forms, the test and the reference product should be administered with a standard volume of fluid, at least 150 mLs. In the US guidance, it says 240. Okay? Point 4, harmonization. It is recommended that water is allowed as desired for 1 hour before and 1 hour after dosage administration, and no food is allowed at least 4 hours post-dose, which is very similar to what the US FDA is recommending. Meals taken after dosing should be standardized, which is pretty much what has been described in the other guidance in the US. For the FET study, standard procedure for food interaction studies, this is what the guidance says. A single dose of the investigational drug, or is it medicinal product, is administered with 240 mLs of water after a 10-hour fasting period and 30 minutes after intake of meal has been stated. Now this I grabbed straight from the guidance. This is a cut and paste from the guidance. The other slide is a cut and paste from the guidance. You see there's some slight differences in the volume and fasting time period. Exception for the meal, the subject should refrain from food at least 4 hours after dosing, and all food intake should be standardized, which is similar across the regions. Now, when a company is conducting a study or a CRO is conducting a study, what do you see? Earlier, I think Anders mentioned that a lot of the things you do in the study you should have in the protocol. And basically here, I'm just giving an example of the plan for a company and what they would plan to do for the whole meal prep or intake of food, whether it's in a FED study or fasting study, when the fluid should be taken, you know, every 1 hour, water and then some juice, and when they should have lunch, snack, supper, and things like that. So this is what you would see in the protocol. And you know, I'm not going to go into too much detail because we're trying to make up time. And this would be what you would see in the study report, what they plan to do and what they actually have done. I wasn't able to find, so this is for a fasting study, I wasn't able to find something similar for a FED study, but then you would have the breakfast line where you would have the high-fat meal and wartime, and then that would be an indication that it's a FED study. So now that we've talked about the guidances, the similarities, and now how administration of the meals during the fasting and FED study, let's talk about the composition of the meals. Under the US FDA guidance for food and food effect studies, the recommendation is that the FED BE study should be conducted using meal conditions that are expected to provide the greatest effect on GI physiology, so that the systemic drug availability is maximally affected. So you're really trying to see an effect as a result of the food. The requirement for the food is a high-fat meal where 50% of the total calories come from the fat, and the total calorie for the whole meal should be between 800 and 1,000 calories, which was also mentioned earlier. The test should derive approximately from each category: protein, carbohydrates, and fat. It lists the calories that should be derived from each food category, and it's listed there. And the caloric breakdown of the test meal should be provided in the report. I know when I was at the US FDA, this is one of the things that you actually, as an assessor, you physically check. If you see the US BE summary tables, they also provide a summary table how you can display the meal so that the assessors can be able to review it. So if the caloric breakdown of the meal is significantly different from the one described above, the sponsor should provide scientific rationale for this difference. For the EU, we see that the specifications are pretty much the same as what US FDA recommends, so I'm not going to go much into the 50% and all of that. So how does the US FDA meal look like? This is basically what has been listed in the guidance. An example of the meal would include 2 eggs fried in butter, 2 strips of bacon, 2 slices of toast with butter, 4 ounces of hash browns, potatoes, carbs, and 8 ounces of whole milk. Now, as Nurifa said earlier, substitution of this type of meal is allowed, especially because, for example, a lot of the studies are done in India. There is a vegetarian situation. However, you have to meet the caloric and requirements based on the composition of protein, carbohydrates, and fat. And I'll give an example in a few minutes. EU standards, pretty much the same. Eggs, bacon, toast, hash browns, and milk. Similar quantities. Same thing, substitution is allowed. However, in the EMA guidance, they also list the moderate meal, and the moderate meal could contain approximately 400 to 500 calories, with fat contributing to 150 calories. I don't know if any of you have done a fed study using a moderate meal, but I know not everyone is doing fed studies, so the chances of finding this example is pretty low. Okay, so now we're at the examples phase, and we're almost done. This is a submission of a meal, and as you can tell, it's different from what has been described in the guidance. You can see there's milk up to 40, which is standard. There's bread, cheese, coriander, jaggery, ketchup, rice flakes, so all of these ingredients that go to this high-fat meal that is recommended still meets the requirements according to the guidance for EMA or US FDA. And if we take into account the proteins, carbohydrates, and all the composition of the meal, we see that the meal still, it still meets the requirements, 150 to 250, 500 to 600 calories for proteins, carbohydrates, and fat, respectively. So these are the percentages, and as you can see, for protein, 153, close to what is recommended. So even though the meal looks different, the composition of the meal looks different, it still meets the requirement. 2 more examples. Here's another example from a different submission. And basically what they've done here is they have the food item, they list the servings and the amounts, and then they also list the breakdown of, how do you make a vegetable cutlet? You need the potatoes, onions, cooking oil, and some cheese. So, if you want to try this type of breakfast, you can even actually do it yourself. But then there's the breakdown of carbohydrates, fats, and proteins, a summary, and this table here shows you we're within the limits as recommended by the agencies. This is just another example. In the interest of time, it's very similar to the recommended meal, except here there's a bacon replacement. We have chicken tikka for protein, and once again, the caloric requirements are met as specified in the guidance. So, our conference is Harmonization of Bioequivalence Requirements. I took the next 2 slides from a publication that is listed in my reference, but this kind of shows what is happening globally, not just in the US, in EMA or GCC, but what are other countries doing. So as it pertains to the different regions, all regions request a single-dose equivalent study under fasting conditions, and most do not request a fed study for IR dosage forms, with the exceptions of below. For example, in Brazil, a fed study is requested when there is a known food interaction. We know the whole story about the US because we just got a whole presentation about that. Singapore, this region, where the SPC specifies. So basically, most countries rely on the labeling of SPC if there's a proven interaction for requesting for the fed study. And that was more for fasting study. Here is a fasting and fed study, and here's the requirement for composition of meals in a fed study. So basically, there's similarities within the jurisdiction, as we can see from the slides that I presented. There are some slight differences, whereas Brazil, China, Mexico, Singapore, they have no recommendation about a meal, so you can choose to they can choose to rely on the other guidances. Chinese, they have a high-fat, high-calorie meal. It doesn't specify the caloric requirement. Japan, here it says 700 calories, of which more than 20% of the energy is from lipids. And, you know, we also have WHO, which just states that the meal should be based on the local customs diet and should be administered 30 minutes prior to dosing. So as you can see with administration and meal composition for fed study, there's a lot of similarities. There are some differences, but not glaring differences, such glaring differences. So at this point, I'm at the end of my presentation. I will be happy to address some questions, or if we're trying to make time, you know, it depends on the moderators.
Thank you very much, Dr. Lois, for this interesting compilation on meals composition and fed study conditions between EMA and US FDA. So we have time for questions.
This is awesome.
Rima Habdadin, I am a freelance monitor. I just have one question about the conduct, during the conduct of the study. If some subjects did not finish or complete their breakfast, shall we exclude them from the study?
If they don't finish I'll answer your question with that question. If they don't finish their breakfast, are they meeting the caloric requirement that's specified in the guidance? No.
No.
So then they would not be consuming the recommended meal, and the ideal course of action is to drop them from the study. I don't know what the practice is in the CRO. Maybe I can get some input from that, but basically when we found a situation like that when I was in the agency, we dropped the subjects. Thank you. And it doesn't really occur as often. This was like one very random situation, you know. And they had many other issues pertaining to their vet study. And so just a good example.
My question is on fat. I think everybody is fat enough for lunch. Maybe I can ask you something.
Sure.
It's related to apple sauce. I mean,
Yes.
What is your opinion? In Europe, there is a question about the dispersed
Yes. So
content of capsules on apple sauce. Can you?
So this is usually known as the sprinkle study, and it's usually recommended for modified release dosage forms. It is in the guidance. It is expected that if in the labeling they say open the capsule and disperse the drug product on applesauce, you should then do a sprinkle study along with your fasting study. I didn't address it in this presentation because this was immediate release, but for modified release product, there are additional requirements that may be needed based on how the product is taken. So you have to take that into consideration.
Don't you think that if a modified release, it's always required in Europe, a fat study?
Yes,
so,
and I think
which is the worst case scenario. So don't you think that an applesauce, which is the lightest case scenario, would be also by the fed state?
I think it goes back to condition of use of the product. So if it's being sprinkled on applesauce and really not taken with a fed meal, then the best way to do the comparison for the test and the reference would be in the conditions of use. At least in my opinion.
May I briefly comment that from our own experience, there's a significant difference in those cases where a capsule, for example, is open and the pellets are mixed under the aerosol. So their GI transit will be different to a normal closed capsule, which is normally opposite to our expectation, not easily opened in the stomach, often swimming on top of the meal. There's no water there, and therefore a significant difference in gastric emptying, etc. Therefore, I believe, I would expect, let me say it this way, I would expect a difference between the closed capsule given after a high-fat American breakfast and sprinkled over the aerosols. So that is not fun. And
Kinetic profiles
sorry.
would be different.
No, that's...
I think the pharmacokinetic profiles would be different.
Yeah, and you asked for experience how to deal with the situation that the meal is not from some control. We normally make a decision at the end, because if a subject is not able to ingest 800 or 1,000 calories, and in those administration periods the situation is same, then we leave such subject in the study. If there's a significant difference, we tend to exclude. And this is in particular a problem in case of patient studies. We do a lot of patient studies, and I had some discussions with US FDA where we disagreed with their position in case of oncologic patients. If you ask them to ingest 1,000 calories and 50% the calories are fat, it is impossible for the majority of these seriously ill patients.
And I think also Nilüfer mentioned that for cancer patients or oncology patients, there is some room for adjusting for the type of patients. Thank you very much for your time and attention.
So now we have Peter again. We also know good morning, Peter. And he will talk about formulation-related reasons for drug-food interactions.
Hello again. Food effects is the third speaker and the food effects, but I can promise you it's not going to be another story, but there is some links between the topics. I was very glad to hear Nilüfer's story about the MCC effect, something that people normally don't realize, and you will see that I have some more mechanistic explanation of what's happening. So, the title is quite broad. It actually this food effect and the formulation related issues I have condensed into a special case, but probably that case is still a little underrepresented, but yet I think it's quite important. And I spent most of my time on it. So, as a response of food effects, I'm looking at the effect of food on two variables. One is disintegration and the other one is dissolution. I'm not looking into more physiological variables, but I hope I can show you that even in vitro investigations can be quite useful in predicting some odd things that happen when you mix a dosage form together with food. And so I spent some time discussing biopharmaceutical implications of those in vitro investigations, and then also share with you some investigations and results we've obtained recently and trying to optimize dosage forms so that they do not show that high effect. Okay, starting with disintegration first. I mean, it sounds a little trivial. You put a tablet in a glass of water or in a beaker and you shake it and you observe whether it's disintegrating or not. But if you look a little bit more careful, then the simple process of tablet disintegration can be quite complicated because there is a lot of elements there that can affect the disintegration process. And you see that here. It's depending on the fluid characteristics of the disintegration fluid, and it's also depending on the hydrodynamics of the movement around water movement around the tablet, and then there are also some mechanical aspects such as mechanical stress that is exerted on the dosage form by the fluid movement around the dosage form, but also directly by contractions of the stomach. The stomach is a muscle, so when it contracts in the fasted state, there is direct pressure exerted on the dosage form. So these factors are important, and if we just look at the fluid characteristics, there is, for example, the viscosity of the fluid that can play a role. There is something that's called water diffusivity, and that is defined as the velocity of water molecules that move within a certain solution, and it makes a difference if you have a sugar solution or a hydrogel or simply a sodium chloride solution. And there is sometimes the effect that food can precipitate out and form a layer around the tablet, like a diffusion layer, and that can create a dosage form to become a controlled release rather than an immediate release tablet. Surfactants can play a role, and then there is some dosage groups, some dosage forms. There is pH and buffer strength effect. That's mainly occurring for enteric-coated controlled release dosage forms, not so much for immediate release dosage forms. Okay, and what are the results of these factors? One is that the water uptake into the dosage form can be influenced, and the other one is that the physical variables can influence the disintegration as such. Water uptake is mainly the first step. Thereafter, the excipients, disintegrants start swelling, other excipients as well, like starch, MCC, and so on. They start swelling, and then eventually the tablet disintegrates into granules or powders if it was a direct compression process. Okay, so that makes us define what we are talking about here. And just to show you a case example, this is a drug called phosphine chloride, which is sold. It's an antimuscarinic drug, BCS class 3, sold on the German market. We have currently 3 products, Spaspi, Spasmex, and Spasmolute. And when we measure disintegration times for these products in different media, we can see that in simulated intestinal fluid, or say in simulated gastric fluid, you have a very nice, very short disintegration time. But as you increase the viscosity of the disintegration fluid, disintegration times really start to go up. This is 0.5% HPMC, this is 2% HPMC, and you can also see that the dosage forms, they behave a little different when you test them under viscous conditions. So I do not agree with the EMA conclusion that the fasted state is really the most discriminative. I rather say the fed state is more discriminative than the fasted state. But that can be debated anyway. So we see with viscosity as an influencing factor that disintegration times can change, and also the mean dissolution time changes. Here's the example Trotsky, which shows a rapid dissolution of the trospium in the simulated intestinal fluid. But if you increase the concentration of the HPMC, for example, to 0.5% and then even to 2%, you can see that this becomes a slow release preparation rather than an immediate release preparation. And the only thing we change is the viscosity of the medium. So we thought a couple of years ago this is quite interesting. And also, if we consider that trospium is a drug which has a preferential absorption in the upper small intestine, as you see here. This is symbolized symbolization of the GI tract. We have the stomach, we have stomach emptying, and then we have a number of compartments here: duodenum, jejunum, ileum, and then the colon. And it is known for this drug that it has a carrier-mediated absorption in the upper small intestine, and is no longer absorbed in the lower parts of the intestine. And that means that anything that retards the dissolution of the compound from its dosage form leads to the lower concentration of the drug in the upper small intestine, and if it's not dissolved, it's not absorbed. So what you would estimate or anticipate is that if your dosage form doesn't disintegrate fast enough, it's not going to be absorbed and you lose bioavailability. And indeed, when that has been tested in humans, in fed and fasted conditions, you can find the differences in the PK profiles as shown here. This is the curve for the fasted state, and down here you see the average curves for the fed state. Same dosage form, just the only difference is fed versus fasted. And the conclusion by the statistics was that you lose 87% of the area if you administer that drug in the fed versus the fasted state. The lines, you might ask yourself, what are the lines? They are in accordance with the simulation model that was developed. It predicted the fasted state based on purely physicochemical and PK profiles of trospium and its permeability and so on. So it was quite well for the fasted state. It over-predicted a bit the food effect, but we know why. There is another component that was not built in the model, that in addition to the viscosity effect on disintegration, affects the absorption of trospium, and that is typically a bile salt interaction. Okay, let's look a little bit more into some mechanistic aspects. I want to show you with some data that our understanding of dosage form disintegration and how the composition of the stomach fluid and the stomach content influences these disintegration times can be visualized. And so we look at the effect of viscosity on water uptake here, and you see the relationship here. We just discussed this upper figure. That is, with increase in the viscosity of my disintegration fluid, the water uptake goes down. Okay, so and it's quite severe that with increase in viscosity, you have less water uptake into your dosage form. And if you have less water uptake, you have less swelling, you have slower swelling, and longer disintegration times. Next, we try to measure and measure the viscosity of the US FDA high-fat meal. And so we homogenized the meal and did some rheograms of that, and then looked for some artificial fluid that would be simulating the US FDA meal, which would be more practical for in vitro studies. Because the US FDA meal, I'm not sure if you have worked with it, in a homogenized state, you can't see anything, not even disintegration of the dosage form. So we needed something else. And to make the story a bit condensed, here's the rheogram. You see here the shear rate versus the shear stress. This is the rheogram of the US FDA meal containing the milk component. And this was here, which came close, at least here, in these higher shear stress conditions, came close to a 1.4% HBMC solution. So we have some deviation down here, but up here it's more or less okay. So we replaced the homogenized FDA meal by 1.4% HBMC solution. If you want the specifications, they are in the references. So if you read the slides, you can get, because it's a question which molecular weight, which substitution degree, and so on. But you can find those information. So all the experiments then are done with 1.4% HBMC solution. Here's an example of how the water diffusivity can influence the water uptake. And we can measure actually the velocity by which water molecules, by an NMR table, by which water molecules can move in a solution. And this gives you a number. That's the water diffusivity. And the bigger that water diffusivity, the higher the water uptake rate into the tablet. So in other words, any components that can be part of the fluid in which the dosage form disintegrates, any interaction that water molecules through hydrogen bonds can have with, for example, sugar molecules, can affect the diffusivity of water, and that can affect the water penetration. Here's a visualization of that. It's an MRI into these trospium chloride tablets. This is trospium, Spasmodex. This is studied from simulated intestinal fluid. And here we have a sucrose solution, 15.8%, so quite high sucrose solution. And you see the time is going from left to right. And this is 180 minutes. Here we have 360 minutes. So we see increased water uptake into the dosage forms, but since they are not identical products, they have differences in water uptake rate. That's one observation. The other one is that we get more water taken up into the tablets when we use simulated intestinal fluid compared to sucrose solution, because here the water diffusivity in this solution is much lower than in SIF. And you can, if you compare like 120 minutes here to 120 minutes here, you can see that water uptake is reduced just by this imaging technique. Looking into some of the test apparatus, how do we measure those disintegration rates? The first test that I found was in the Swiss Pharmacopoeia. There in the edition of 1933, there was, for me, the first disintegration tester for tablets published, and they say that you should take a 100 milliliter Erlenmeyer flask, you add 50 mils of water, temper you warm it to 37 degrees, and the tablet should disintegrate to a powder within 15 minutes. Okay, that was the initial start before any dissolution test was run. Today we are still in a rather primitive fashion that, okay, we have a pharmacopoeial instrument, but it has several disadvantages. I mean, you know the instrument, if you've seen it, the basket rack assembly, you have these tubes here, you can put one tablet per tube, 6 per rack. There's a mesh on here on which the tablet is positioned, and sometimes you can run it with discs to avoid the swimming of the tablets on the water or fluid, whatever it is. Disadvantages is this has quite a high fluid velocity in this instrument. You cannot adjust it. The instrument moves 30 times per minute up and down. It's limited to a sinusoidal profile, no mechanical pressure, and a limitation to vertical movement. So we have used computational fluid dynamics to quantify the fluid movement in this disintegration tester and compare different fluids like gastric fluid, simulated gastric fluid, HBMC, and the FDA meal. And you can see just by looking at the colors that the fluid of HBMC and FDA meal are quite similar, same colors, whereas the velocity of the SGF is much higher, causing more hydrodynamic stress. And using this simulation, we can also calculate the stress that is exerted from this movement on the surface of the tablets on the FDA meal as well as the SGF. So we know under which stress the I wanted to show you a movie, but it's not working. Okay, what I tried to show you in the movie was a better instrument, right? That would be adjustable in terms of hydrodynamics, and it would have a balloon attached to it, so you can directly simulate the effect of gastric contraction on the dosage form. And it can move in 3 dimensions, so it can do any movement, any velocity, and we are sure you can more easily adapt the physiological conditions of the stomach than using the old disintegration tester. This picture shows you a relationship between the 3 variables. The depending variable, that's the disintegration time, the average shear stress that we calculate based on CFD measurements, and then the velocity of the new disintegration tester. And we used it for 2 tablets, same composition but different compression strengths. And you can see how nicely the predictions are, or the simulations are, for the experimental result. The longer the disintegration time, that means that the average shear stress is lower and that the fluid velocity is also lower. So fluid velocity and average shear stress determine the disintegration time in such an instrument, and that depends, of course, on the type of tablet. So you can basically use CFD if you know fluid velocity, if you know viscosity. You can use CFD simulations to predict disintegration times. This not just holds true for trospium chloride tablets, but we have looked at several others, and we found some interesting results. These are products bought from the market, tested under low and high viscosity conditions for disintegration times. And you see A1 is atenolol, A1, A2 are 2 atenolol products. This is fasted state fluid, and this is under high viscosity conditions. So the interesting thing is, of course, the viscosity has a negative influence on disintegration time, but then it depends very much on the fluid composition, on the tablet composition, if you have this as a large effect or as a lower effect. And it also holds true for furosemide, for example. Here, F1, F2 is furosemide product 1 and 2. Also, you see the same effect. But it doesn't mean that all products are very sensitive to viscosity conditions. This is, for example, metformin, and there we did not see any of these effects. And here is also metoprolol, which didn't seem to have that effect. So that made us then think, what should we in addition want to know? And that was the question, how are the process parameters and the components in the tablet affecting the disintegration times under fasted and fed conditions? And so here are some slides with respect to this investigation. We have here lactose-based tablets using phosphine chloride as an API and tested under fasted and fed conditions. You see, when we have no disintegrant, in the fasted state we have relatively higher disintegration times. The difference here is the compression force. This is 10 kilonewton and this is 30 kilonewton. But if we add super disintegrants, either goscarmelose or gospropicolone or sodium starch glucolide, this disintegration time drops quite dramatically. And this is typically what you observe in the normal studies, because all of the studies are done under low viscous conditions. Super disintegrants work, okay? But if you study them under high viscous FDA conditions, you see that they do not work very well. This is no disintegrant, and here's the effect of the so-called super disintegrants. And they do some of the job, but no comparison to fasted state conditions. So the testing as well as the tablet composition seems to be important. And this is not just reflected in the disintegration time, but also in the dissolution times. You see these here under fasted condition, the dissolution of osmium with these different super disintegrants, and this is the fed state. You can just, I don't have to give you numbers, just by looking at it you see that the dissolution is retarded. We then started to model this to find a more mechanistic explanation for what's happening there, and we took these force time curves. That is, we take the tablet, we immerse it in water, and we measure the force that is exerted upon expansion of the tablet. And this is a force time curve, and you see at the beginning there is no force. The force is rising up to a maximum force and then is decreasing. And we define then the maximum force at the Tmax of that force. We define a disintegration force development rate, which is the slope here, and we define a disintegration force reduction rate, which is HBR, which is this slope here. And if we use that kind of analysis, we can group the tablets and their formulations into their different categories. And that is shown here, just as an example. For the fasted state, we have these typical force time curves. If you look at the scale, this is time in seconds. And here we have it, the same tablets in the fed state. And you also see, this is time in tablet, this is time in seconds, but the numbers are very different. This is 20 seconds, and this is 900 seconds. So for the fasted state, where we have fast disintegration, we have low Fmax, and we have a very fast disintegration force development rate. So very high slopes here. And that can be easily explained because water is going into the tablet very quickly, and then the tablet is disintegrating, and then the force is gone. So the curves must be like that. Whereas if you are in the fed state, it takes much longer time for the force to develop, and still here the tablet is not disintegrating, and it starts slowly thereafter. Finally, how much time is still there? I'm not sure if 3 minutes? 5 minutes? Okay, thank you. So I can probably finish this part. We did a more complicated design of experiment study using different disintegrant combinations. We wanted to know which of the combination of disintegrants is best and least affected by the viscosity. And we took them at 2 levels. So this is phosphoridone, phosphorylose sodium, and then sodium starch gluconate, and at different combinations in terms of concentration and in terms of combination. So the low level is 4%, and the high level is 8%, so 4% each. And we also looked at the effect of the filler, whether it's lactose, soluble, water soluble, or water insoluble decalcium phosphate, DCP, or microcrystalline cellulose. And here's the bridge towards Nilophar. In a Nilophar's example. Other excipients are PVP-K30, magnesium stearate, this is a binder, this is a lubricant, and this is the drug, again. So, this is the disintegration times you see here in fasted state media. I should explain a little bit. So this is the dependent variable. Here we have the composition. This is, for example, lactose at 10 kilonewton, at 30 kilonewton. This is DCP at 10 and 30 kilonewton, and this is MCC, 10 and 30 kilonewton. And you see these blue points here. These are formulations that did not contain any disintegrant. That's one thing. Disintegration, as we saw before, in fasted state media matters, if you have a disintegrant or not. Second observation, it works quite well for both soluble as well as insoluble fillers. If you test it in fasted state media, the effect of super disintegrants is obviously there. But here, when you look at MCC, you can already see a tendency towards higher variability depending on the composition of the tablet, and a generally higher disintegration time compared to the other recipients. And it is even more interesting when you look in the fat state. There we have the following observations. Lactose, in the presence and absence of disintegrants, seems to be quite okay. The variability, of course, is higher than in the fasted state media. For DCP, we get even a more spread. Nevertheless, it was also clear that DCP disintegrates faster if we use the higher compression force, 30 kilonewton versus 10 kilonewton. And we have some mechanistic understanding why that is so. And look here, the MCC. Most of the numbers are, in terms of disintegration, are more than 45 minutes. This is where we stopped the experiment. And so this was quite an interesting observation that we thought. So MCC is, and I jump over the dissolution curves, they show you the same, come to the conclusions. Well, it's not trivial to formulate a tablet so that it doesn't show a food effect under enhanced viscosity conditions. And the negative effect of food-induced viscosity on disintegration and dissolution cannot completely be avoided, but you can try to minimize that effect, for example, by choosing a combination of phosphorylose sodium and phosphoridone in lower concentration. You don't need more than 4% of the compression away. When DCP comprises the major proportion of the tablet formulation, we recommend to use higher compression forces. And please forget MCC, because this is in all studies, and I'm very glad Nilophar has brought that example. Even there, we saw that high concentrations of MCC seem to retard the release and the disintegration from the tablet. With that, I'm done. Thank you for listening. I'm looking forward to this.
Thank you very much, Peter, once again, for revising this dynamics and chemistry of simple disintegration studies, which are not so simple as we might think. So we have time for questions.
Thank you for the presentation. Again, Hakima from Hikma Pharmaceuticals. My question is, how can I relate this disintegration and the effect of exceedance with the dissolution later if we conduct the dissolution? You know, at the time, the disintegration was one of the critical points upon study quality parameter, and then we have to change to the solution. In bio and bioequivalence, how can we relate for that? Thank you.
I didn't show you the pictures, but we have correlations of disintegration and dissolution numbers. For example, time for 50% of the dose to dissolve, or 75%, and mean disintegration times. And you find, if you look into different products that have different disintegration and dissolution times, you can regress this and find significant correlations between these variables. So there is definitely a relationship between disintegration and dissolution, I would say the difficulty sometimes with the disintegration is that you need to identify and define a good endpoint. So you can say, okay, is it completely disintegrated when nothing is left on my mesh? Yeah? Or is it disintegrated when it falls into granules already? So that is something, but we use high-speed cameras for that kind of, we film the whole thing for each tablet, and we can then very clearly define the process for each product. And if we take these times, for example, which is not trivial also in the case of entry-coded dosage forms, if you define as a disintegration time the time it takes for the whole product to leave the mesh, that's much too long. So that when the first crack, for example, starts, then drug is going to be released from it. And that you can correlate with dissolution parameters, and you find it works out quite well. So these two in vitro go along quite well. You can even use one to predict the other. This is not for regulatory purpose, but we have found you can use it. And then I think where we're still lacking examples is on the in vivo relevance of delayed and prolonged in vivo disintegration times and how it relates to pharmacokinetics. That's why I'm so fond of Nilofar's example, but we have also other data from dog studies where we can show that this is also happening. So we've taken multiple X-rays in dogs, for example, and we can define in vivo disintegration times and relate that to the in vitro testing. Does this answer your question? Or yeah. That's, of course, that's more expensive to do those kind of studies. And you need imaging techniques and so on. Or you need good pharmacokinetic studies. That's the validation part, and that's the harder part. Yeah, definitely. Yeah. There is examples out there, not just from us, but from literature as well.
One last question, Dr. Mohamed.
Yeah, very much, Prof, for the interesting presentation. A couple of points actually. As a regulator, yeah, definitely disintegration is linked to the dissolution, and bioregulation says if there is a dissolution requirement of the finished product specification, you don't need to have a disintegration testing parameter because they call CQA, critical quality attributes. And in this case, the professor work is interesting, assuming that it could be proven that this is a CQA that based on what he presented as to satisfaction of the regulation. I know you said this is not for regulator, but I mean, for us, we think as a regulator to consider a disintegration as an indication for the quality of the product, I need to have that sort of link. That's one point. The other point about the shelf life. how we test that disintegration will withstand the storage and to be as reflective as a dissolution. So we'd like to see that disintegration test is actually will be a guarantor to the quality of the product over shelf life. And that would be very interesting. So it is very nice because I'll tell you, I have seen an EMA, we call it EMA, I don't know, you call it EMA, like a name, we call it EMA. EMA, we have seen that one of the centralized products was actually approved based on disintegration, not on dissolution. Because they thought that disintegration, I can't remember now exactly the name, was that of 2013 or 14, and was actually approved based on disintegration, centralized application, but not on dissolution. And that was very interesting because usually the process of disintegration will be followed by dissolution. And in this case, the disintegration will be the faster stage compared to the dissolution. The disintegration takes about, for immediate release, although in the monograph say 30 minutes, but most of the work that we do as a development, 5 minutes, and then we'll allow a certain time for the storage to be up to 10 minutes. But the dissolution takes up to 45 minutes. So this time, lag time, I hope the professor will explain that to us. Thank you very much.
Yeah, thank you. I fully agree, and I think, I may be wrong, I'm not a regulator, but for BCS class 3 substances, I think those that are very highly soluble, in some cases you can use a disintegration test because it's all happening before the gastric emptying time. So it's a very quick process. But my focus was to put some evaluation and some energy into a neglected process. That it may have a mechanistic explanation for some of the events that we don't know how they happen.
Peter. Thank you very much, Peter. So we will follow with next presentation with Dr. Muharrem. Necessity of food effect studies in case of modified release products.
Hello, good evening. Can you hear me then? Yeah, all right. Just to tell you, the presentation is all about regulation, what the EU would think about the food effect, or about the modified release equation. This is extracted from the guideline published in 2014. I know FDA just published in February 2019 a new guideline about for new drug application and for the generic. But this is in 2014, so we are ahead of the FDA. Anyway, so what I'm going to discuss about here, about the food effect, the biology of drug substances, BCSE class, what do we want for the modified release product, how do we define a generic modified release product. This is very important. Then the food effect and all the following subject. Of course, the administration of drug product with food may affect the bioavailability, bioavailability. Either the affecting the drug substance itself or the drug substance. And in this case, as the professor said, that you have to, for exact mechanistic studies and specific mechanistic studies. For BCSE class, which is highly soluble, highly permeable, nothing will stop in front of them. They will go through regardless because they are pH and site independent. And that's why you don't expect anything will actually will affect the dissolution of BCAC class, and hopefully will then be insensitive to any changes. However, there are certain things that we have to consider, and I'll tell you an example about. For example, there are other factors have to be taken, even for the BCAC class 1. For example, a high fast-pass metabolism effect, extensive absorption, complexation, or instability of the drug substance in the GI tract. I'll tell you, this is a guideline, and we'll explain these things. But sometimes, as a reader or as a working pharmaceutical industry, you don't really pay much attention to what the regulation is saying, because it's actually mentioned in a smaller sentence. But sometimes, believe me, that will affect all your work that's been done over the last 4 or 5 years, because you haven't read that sentence. And I'll tell you about for that first pass metabolism effect. Now we are dealing with a product as a regulator for an oral solution that's not been licensed before. There's only an immediate release preparation. The immediate release preparation, the drug substance is well known to have 85% is actually subject to first pass metabolism. So the assessor asked about the safety and efficacy of the oral solution of a BCAC, and the BCAC, of course. He asked for the BCAC class and what would be the implication of having oral solution compared to the IR tablet regarding the safety and efficacy of the proposed product, since there is 85% of first pass metabolism. The idea that the IR tablet will be slower than the oral solution, because it takes disintegration, then the solution, while the oral solution will be already there. So the first pass metabolism with the oral solution, it might be faster, and then hopefully the PK profile will be different, and in this case, where is the safety and efficacy? And this is what we are dealing with. So we met MHRA, which was actually strange, because I was sitting as a consultant, not as a regulator I used to be. So my colleagues started to sit opposite to me, and we agreed to do the following studies. First of all, to decide the BCAC class of the drug substance, which is very important to make sure actually it is BCAC1, as we have good evidence that could be BCAC1, but we are not sure because we don't have the actual experimental details or data to prove that this is BCAC1. And the other thing we agreed that to do the solution study of the IR tablets, because it's already licensed, and hopefully the idea is actually to show that the IR tablet is so fast in dissolving, because it's BCAC1, and in this case, any different, and in this case the dissolution process, or the discretion will all occur in the stomach before it gets emptying, and then any first pass effect will be similar for both dosage forms. This is the strategy that we agreed with MHRA, and he said in this case, all right guys, go and do it, and we are still actually in the process of working on we've done the BCAC1, and quite happy to say that we have established that it is BCAC1. We're still working on the dissolution of the IR, and hopefully we'll gather all that, and then to pharmacokinetic bridge of between two products to be submitted to MHRA soon. And of course, if you have any idea to improve what we are doing, well, always thank you very much. So one of the other things is, which I'm not 100% still have an answer to it, is the effect of caffeine on the absorption of drug substance. Last time, when I was with MHRA, we had a meeting with the top-class pharmacokinetist in the world, Professor Malcolm Rowland, and other three, and we all discussed about the caffeine effect and the effect of the caffeine on the gastric emptying time. Actually, it wasn't clear exactly where we stand, and I looked everywhere to see if there is anything that will tell us about the caffeine effect and with regard to the BCAC class. This has not been covered in any guideline. Anyway, this is just something that I'm thinking about. Right. Of course, excipient as the professor just showed, and I think we are studying that it might influence the Cmax and Tmax by delaying the gastric emptying time and the prolonged intestinal transit time. However, these factors, when we come to the generic or to the test product compared to the reference, all these are actually will be balanced off. That means what happened to the reference product will also happen to the generic, so nothing to worry about if you're going to go to do the bioequivalence studies as designed. Right, for the other IR drugs, which is for BCSC class 2, 3, and 4, as well as modified release, the food effect is more complex. And that will affect in vivo dissolution by nature of formulation. And of course, once the dissolution will be affected, then the second stage will be the absorption of the drug substance. And in this case, we have to study the magnitude of field effect and demonstration of bioequivalence. Here are the guidelines that I think has been referred to during the previous talks. This is the main skeleton EU directive. Actually, it's almost like our, we have to read it every day, twice a day before food, because you have to understand each word of it, and that will be the EMA, that before it becomes EMA, it was EME in 2010, and then followed by the guideline on the pharmacokinetics and clinical evaluation of modified release in November 2014. What do we need to define the modified release is actually by determining the, just like the immediate release, the Cmax as a rate, and the AUC for extent. The other thing we need to see is the fluctuation in drug concentration at a steady state. And if there's any intersubject variability in pharmacokinetic arising from the drug formulation, and of course, if those proportionality will be applied. To define generic medicine, this is the definition that comes in the guideline 2010. It should be chemically equivalent. By the way, the guideline for 2010 of the EU is only about immediate release. So this is actually copied from immediate release, and then try to discuss where would that fit on the modified release. Chemically equivalent for the immediate release, they consider any salt, any isomer, any type of product of the same active substance could be considered as chemically equivalent. Then they have to be pharmaceutically equivalent. And they consider any immediate release dosage form, like capsule, tablet, oral solution, are all the same, are all considered one. And in this case, if you have an immediate release capsule form, and you want to design or develop an immediate release tablet, by all means, there is no problem with that, because this is all considered, especially when you refer to the reference standard. If you have a tablet, you can refer to a capsule, there's no problem with it. But will that apply to modified release? No. Because if it is a tablet, it has to be a tablet. If it is a capsule, it has to be a capsule. You cannot interchange. When you refer to the modified release, it should be the same dosage form. And of course, for generic medicine, it has to be bioequivalent to claim for all the safety and the clinical studies that we carried out for the originator. Of course, how do we define the reference product for the modified? Any product, including the modified, it should be either the originator, which is the originator modified release that has been approved by EU, or a line extension of an immediate release by the same applicant. And all these is actually according to the 2010 guideline. What I'm going to talk today is only about modified release. This is a very crucial slide It says if the two products with the same dosage form differ in their release controlling excipient and or mechanism, only consider generics if bioequivalent in vivo after single dose in fasted and fed state. So the guideline is actually didn't give you the choice whether you go for fasted or fed. It has to be in both if you are a generic or modified. I hope that will be clear. There is no shortcut in that. You have to have the bioequivalent, fasted and fed for the two products. And of course, you might need later on multiple dosing if there's any accumulation. We'll go through this quickly. The idea of the why do we need bipolar studies, not only actually to show is exactly the same as the reference product, which is the ultimate results. One of the things is actually we want to see the claimed prolonged release characteristic. By the way, I forget to mention something, it's not in the slide. How do we define from immediate release of the modified? I have seen it actually in many forum discussions. Sometimes they forget the definition. I've seen it in many applications from the wholesale industry. They forget the definition of the immediate release product. For us, in the European pharmacopoeia, we define the immediate release product that it will release 75% of its content within no more than 45 minutes. And that's why you have Q value. All right, in the hospital. You have a Q value because within 45 minutes, not less than 75% will be released. For the modified release, anything that takes more than 45 minutes or 75 will actually need 2 points of estimation. And that's how we define it modified. Is that clear? But this is very important. I have seen application, someone is actually having the Q value at 60 minutes. Because the 60 minutes is actually outside the definition of the immediate release product by the European Pharmacopoeia. So when you get that done, is actually what we're getting is less than 75 within 45 minutes to get it modified. So in this case, it has to have a prolonged release characteristic of the reference. The active substance is not unexpectedly released. There is no dumping effect. And in this case, they are equivalent at a single dose and at a steady state. And as well, the food effect as we go for comparable formulation. If there is any difference, look this slide. If there is a difference in food effect between the generic or the test product from the reference, it is no longer defined as a generic. All right? So in this case, if you do the food study, you've done the FASL study and it was all right, then you go into food fed state, and there are differences between your product and the reference product on the fed state. From the EU point of view, they don't consider it as a generic. Under 10A article of the guideline, 10.1, a generic guideline, close of the article, but you can go into 10.3. 10.3 is the hybrid, and that you have to have a clinical justification of the possible effect of the food on the safety and efficacy. And in this case, you get more into the clinical side rather than into the pharmaceutical side. And I think my previous colleagues have spoken about the type administration. This is a very table will show you what sort of type of food. And I'll tell you, this is what it says in the FDA guideline. I copied that from the guideline FDA because it talks about two eggs fried in butter, and then 2 beef sausages. I put 2 beef sausages because they put there as 2 bacon. It might not be possible for this kind of work, so I put 2 beef sausages and 2 slices of toast with butter. And that's sort of high meal, as an example. The other thing which I wanted to talk about, although this is not a food effect, but this is something of the pharmaceutical development requirement that required as well through the compilation of the dossier and submission, is the alcohol effect. The alcohol effect actually being brought to the attention of the authorities, I think by the Dutch authority, who realized that for opioid preparations, people who are actually taking opioid, and some of them, they are heavy drinkers, they have seen some toxicological effects. Then they actually spread a letter to say, we need to control the alcohol. Anyway, after long discussions, it's been agreed, and now in the guideline, to say that you have to test under 0% alcohol, 10, 20, 30, and 40%. One of the arguments that was actually in a modified release conference held every year in London, the chair of the conference said, what do you mean by 40% alcohol? This is a whole bottle of vodka. The idea is actually just to see how far you can go with your formulation and how can you withstand the alcohol, because believe it or not, most of the formulation are actually will lead to dose dumping with alcohol. And in this case, this is very important. So, of course, there is something about what do you do if there is alcohol effect on the reference product as well. Because you are as a generic, you check that on your product, and you have to check it on the modified release as well on the reference product. And if the effect for both is the same, does not justify you to say that actually, okay, I like the reference product. You still have to actually to give a good opinion and justification of what would your product do in case of alcohol effect. Before, we used to accept that actually to say in the SPC, avoid alcohol intake. This now is no longer acceptable, but has to be justified of clinical significance. These studies, of course, you have to do the single dose fasting and single dose meta study and multiple dose study if required. These are the design of the bioequivalence study recommended by the guidelines. These are not mine, these are the guidelines. To say if you want to do a passive and steady state for a single dose study of the test product versus the reference, this is what they give you. And I draw that certain type of them. Before we go into this, I would certainly advise you not to actually, I think my colleague in the Portuguese authority, I advise you, I highly recommend that if you want to design any bioequivalence study for single and steady state, please go and meet the authority and have all your thinking put forward to the authority. They call it scientific advice meeting. It doesn't cost a lot other than the ticket of the airport, of the flight. You go and sit with the authority to agree on which study to do. I have seen a company have actually spent £800,000 on steady state for their modified release products. Yet, and they have 3 scientific advice meetings with the German authority, and at the end, they were in trouble. That's why they come to me. So, don't take, this said in here on the guideline that you can do these or those studies. I think the last one is what we usually do, direct, straightforward. But, so, go and see the authorities. I don't go through these. If we have multiple dose, then in this case we go for the high strength. How do you define accumulation from the literature review? If the first dose of the mean AUC 0 to t time is actually covers more than 90% of the mean AUC from 0 to infinity. That means most of it has already been released. And in this case, there is no accumulation. And in this case, you don't need a multiple dose. You only need a single dose. If you fail, by the way, this criteria for the AUC will be applicable for both reference and test. Don't come and tell me that mine passed and the reference failed. Because the reference is the reference and you are copying. And in this case, the guideline saying both test and reference. If the reference failed with this criteria, you have to go and do multiple studies, despite the fact that your product actually agrees with the criteria. So it has to be both. All right? So in this case, we have to do multiple study, and for the steady state with the food intake, we just have to rush. So one of the main point about what do you do by multiple studies, because if you do multiple to get to the steady state, then you have to think about the washout period, because it will be steady state and it will take about what? I don't know, it depends on the product, to come down before you go to the second stage. And in this case, this point has to be taken into consideration. Usually, it will be 5 times the half-life, the terminal half-life. The other point, which actually I highlighted as in bold, comparing at least 3 doses. When you come to the steady state, the guideline asks the sponsor or the company to show that the last 3 pre-doses are actually comparable. It doesn't say how you compare it. Katarina, do you know about this? How do we compare the last 3 pre-doses? I was stunned actually to say that these 3 pre-doses has to be compared exactly the same criteria as the 90% confidence interval between 80 and 125. In other words, if you have 3 steady state points at the end of the study, you have to compare your 3 points against the reference, and you have to apply the rule of 90% confidence interval 80 over 125. If anyone failed, then in this case, your study is actually a failure. And honestly, I don't know how the formulator can tell what can do for the formulation to make sure actually the last 3 pre-doses are actually complying with the requirement. This is very hard. The company came to me and I found a very good gap to go through and all approved by this. There is another way to call it, SPC1. Anyway, how many times? How do we still have time? That's it? Thank you. I wish I could go for the bioequivalence strength because this is much more complicated. Thank you very much.
Thank you very much. We are 2 hours late, so maybe one first question. Or at least if you have questions, Doctor, you will be totally available to discuss with the attendees. Okay, thank you very much.
I really like that situation. It's 6 o'clock, that's end of this afternoon. We have a general discussion right now on the program. We have a coffee break, and we have another session. I think it's impossible to meet these requirements, and therefore I would like to ask you to go for a coffee break, and because we have to talk to the speakers of the next session, whether they would accept that it was postponed for tomorrow. I think that's the only chance. As all of you very disappointed about that because discussion is important, but time is running. And to be frank, in the morning was not the responsibility of the speakers. It was, as you know, the late beginning of the opening and etc. And therefore we have this problem. So I cannot announce something without speaking to the speakers, and therefore I would like you to go out for a coffee, and we will inform you there.